Hello, my name is Kambiz Hamadani, and today I'll be talking about the enzymatic mechanism of DNA polymerases. The learning objectives for this talk are to show you the structure and various domains of DNA polymerases. So the finger, thumb, palm, and exonuclease domains specifically. We'll talk about how high fidelity for polymerization is achieved by these enzymes. We'll also talk about the transition state stabilization of the nucleotidal transfer reaction and how divalent metal ions and aspartic acid residues function in that transition state. Finally, we'll talk about how the energy released during the reaction cycle is used for proofreading and translocation. So the structure of DNA polymerase enzymes actually resembles very much a right hand of sorts. So the domains are named as such. So we have the thumb domain over here in red, fingers domain in blue, palm domain in yellow, and there's also a wrist, which is the exonuclease domain down below in white. Here we see the bound form of DNA polymerase with the template and newly synthesized DNA strands. The newly synthesized strand is actually making an excursion down to the exonuclease domain in the structure that's shown here. The active site residues that are catalyzing all the reactions, adding the new nucleotide to the growing strand, are positioned here in the interface between the fingers, thumb, and palm domains. The actual chemistry that is occurring is shown here at right. We have a 3' OH group that will serve as a nucleophile attacking the positively charged phosphate, the alpha phosphate of the DNTP that is positioned in the active site and which is hopefully making complete Watson-Crick base pairing interactions with its complementary base in the template strand. As with most highly processive enzymes, in order to carry out the bond making and breaking, the chemistry that I just described, DNA polymerase actually cycles through a number of different conformational states, as we see here. So binding of DNA induces a change in the energy landscape, which favors essentially the population of an intermediate state shown as this yellow state that is not really very highly populated in the unbound form. Once nucleotide binds into the active site, the proper nucleotide, one of these three conformational substates is going to be further stabilized. And what we see is that the cycling of the DNA polymerase enzyme through these various intermediates with two major conformational changes that are occurring as in each reaction cycle essentially helps to facilitate various aspects of its function. So let's take a closer look at the conformational changes that are occurring upon nucleotide binding. So once a nucleotide binds into the active site, if there is a correct nucleotide that gets bound, then there will be a conformational change that occurs in which this helix from the finger domain swings down into position and locks the DNTP substrate into the active site. Now, if a DNTP that is not making proper Watson-Crick base pairing interactions with the template gets positioned in this spot, the helix will essentially not be able to close down. There's also an additional check on the correctness of the Watson-Crick base pairing, and that comes from two residues within the enzyme's active site, which are going to be sensing hydrogen bond acceptor groups in the minor groove of what will become the double-stranded DNA that will be made. So the minor groove of DNA has two hydrogen bond accepting groups, irregardless of what the base pair is in that position in the DNA. So whether you've got an AT or a GC or a CG or a TA, you're going to have two hydrogen bond acceptors in that minor group. And so what this means is that these residues are going to be able to function for all base pairs as long as they're Watson and Crick base pairs. These residues will be able to sense that the correct Watson and Crick base pairing is occurring, and they will be able to stabilize the conformational state with this helix locked in the downward position, essentially the induced fit conformation that tells the enzyme, hey, we're ready to rock and roll, go ahead and carry out that nucleotidal transfer. Right. So once the induced fit conformational change has occurred, and the helix of the finger domain has docked onto the pair of nucleotides that are going to be base paired, or that are base paired, the transition state is going to be fully stabilized by a series of magnesium ions, 1, 2, and 3, together with some coordinating aspartic acid residues and some other residues in the active site here. And these are going to function to do essentially two things. The first is they will turn the OH group on that 3' end of the growing strand into a super strong nucleophile by promoting the deprotonation of that OH group into an alkoxide. So this occurs mainly through interactions with a very positively charged magnesium ion that is very close to it. So this magnesium ion will lower the pKa of that OH group dramatically, allowing it to deprotonate and turn into a super strong nucleophile. The other thing the magnesium ions are going to do is to stabilize the negative charge that builds up in the transition state on the pyrophosphate leaving group, as well as on the alpha phosphate that is the electrophile in this reaction. So those magnesium, the positively charged magnesium ions, or sometimes other divalent metal ions, are going to essentially suck away some electron density from those groups. All of this is happening only when the correct nucleotide has come in and made proper Watson-Crick base pairing in the active site. Once the main catalytic event has occurred, the phosphoanhydride bond that has been hydrolyzed between the alpha and beta phosphate groups there is going to release a tremendous amount of free energy. And that free energy is going to be used in the next step, which involves a conformational change of the enzyme, in which the growing strand is going to essentially get booted out of the active site by all that energy that's been released, and make an excursion into the exonuclease site. Now, if it stays there for a very long time, the nucleotide that was just added is going to get cleaved off. However, if the growing strand zips back into the template as a result of being the correct nucleotide being added and correct Watson-Crick base pairing being possible, then the growing strand will just continue at the next step of the polymerization reaction. The final step in the reaction cycle of this enzyme involves the translocation of the DNA polymerase along the template to the next base that it needs to decode and find a match for. And this last step is going to also be facilitated by the energy that is going to be released as a result of the pyrophosphate leaving group basically exiting the enzyme's active site and ultimately being cleaved into two inorganic phosphates that ultimately will give us the energy of that from that phosphoanhydride bond. So this the energy that's released is essentially going to drive the physical translocation of the enzyme along its track. And this occurs, and it's thought that this occurs via a ratcheting mechanism.
So here we see the reaction cycle of DNA polymerase with the finger domain making the induced fit conformational change, bringing in nucleoside triphosphates into the active site. When we zoom in on the active site, we can see how the finger domain is bringing in those nucleotides and sandwiching them in place, hydrogen bonding and transition state stabilization, allowing the nucleotidal transfer reaction to occur at each elongation step. Here we see the transition to the exonuclease domain, where the conformational change allows for the excision of the erroneous nucleotide and the translocation event to examine the next nucleotide in the sequence.
And here we have an example of a poll everywhere question that I typically use to assess students' learning in lectures. I also oftentimes will connect what we learn in lecture to cutting edge research that's being done in the field or that's related to the research that I do in my lab. So here we see a single molecule FRET assay that is designed to track the various conformational changes that were described in the lecture that I just gave. So the placement of dyes at certain positions allows us to see the shape shifting of the enzyme as it goes from open to closed during the initial induced fit conformational change that is required for transition state stabilization. And then the second is a FRET assay that's designed to track the excursion of the growing strand into the exonuclease site during the proofreading stage of the reaction cycle.